Welcome today to today's Lipid Maps live webinar, which is going to be presented live from Nashville by Professor Alan Brash. My name is Valerie O'Donnell. I'm delighted to be introducing Alan from Cardiff University in the UK. This webinar has been recorded, and as usual it'll be uploaded to our YouTube channel. It's sponsored today by Cayman Chemical, who are specialists in the manufacture of mass spectrometry standards, lipid standards. So during the webinar, feel free to use either the question and answer button or the chat box for questions. Ideally, write them in as you think of them so that at the end we can unmute you and you can ask your question of Alan yourself. So to introduce Alan, Alan Brash grew up in Scotland, and as you'll hear from his accent, he's been in the States for a long time, but he still has very much a lovely Scottish accent. He received his BA degree in medical sciences from Downing College, Cambridge, his PhD on the analysis of prostaglandins from the University of Edinburgh, and then he undertook a fellowship in clinical pharmacology at the Royal Postgraduate Medical School in Hammersmith in London. Following that, he moved to Vanderbilt University, where he has been for many years Professor of Pharmacology. Alan's an analytical biochemist who's had a long-standing interest in lipoxygenases and cyclooxygenases, particularly focusing on the enzymatic and non-enzymatic transformations of hydroperoxides. He has a huge interest and a huge knowledge base on lipoxygenase reactions, which are his prime speciality. We're delighted today that he's able to come and talk about lipoxygenases, really important pathways involved in multiple biological processes in health and disease, both in animals and in plants. And this is Alan's main area of interest, particularly regarding the control of the stereochemistry of the lipids that these really interesting enzymes generate. It's an interest he's developed over the last 20 years, particularly unravelling the role of essential fatty acids in the 12-OR-LOX pathway in forming the mammalian skin permeability barrier. I've been honoured to have had the chance to collaborate with Alan a number of times in the last 10 years, and Alan is one of those people, there are some people who, that if you convince them of something that you're probably right, because their bar is so high that you respect the views that they have so well that if they've considered that you've done what you have to prove something, then you know that you're on the right track. And Alan is one of those rare people who I always use as my sort of bar of, am I on the right track with experiments that I'm doing? I'm proud to say that he's been a collaborator of mine for a number of years, particularly in relation to some work that we did 10 years ago when Chris Thomas, a research fellow in my lab with an interest in skin lipids, won a Marie Curie fellowship, and spent 2 years in Nashville characterizing large numbers of oxidized ceramides with Alan. And since then, we've continued to collaborate a little bit on psoriasis studies and platelet lipids and structural characterization. Alan's seminal work on lipoxygenases in skin spans a huge number of rigorous analytical publications, far too many to mention, some of which he's going to be introducing today. And I just want to highlight that working with Masashi Akiyama in Nagoya last year, Alan demonstrated that short-chain dehydrogenase reductase family 9C member 7, abbreviated to SDR9C7, is responsible for oxidation of a critical lipid required for formation of the skin barrier. This is a really important finding because formation of the skin barrier is essential for life, and the mutation in this gene results in a very unpleasant disease called ichthyosis. And this seminal finding was published last year in JCI, and I think Alan's going to mention this towards the end of his talk. And this kind of work, identifying the underpinning mechanisms and the biology and the biochemistry of lipoxygenases, is really important because some of these pathways are so important in health and disease, and the work that we do to understand the stereochemistry of the lipids is really the underpinning work that underpins all those clinically translational studies in this area. So I'm delighted today to introduce Alan. I've been a big fan of his work for many years, delighted to collaborate with him, and really looking forward to hearing his talk. And I'm delighted to hand over to Alan now.
Great. Well, thank you very much. Very generous introduction, and thank you for the invitation to speak today. Right, lipoxygenases, insights into enzymatic and biological mechanisms, at least from my point of view. Here's a short outline of where we're going. A little bit of background to begin with, then something about the functioning of these enzymes in very general terms. Then I'm going to dig into 3 different aspects of the positional and stereo control by these enzymes in making their products, the fatty acid hydroperoxides. And as part of that, go over 4 lines of interest I've had in the R-specific lipoxygenases, as we'll see. Then get back to the R versus S stereo control of product synthesis by these enzymes. And finally, I'm going to give a short overview of our project looking at the role of the 12R lipoxygenase pathway in forming the mammalian skin permeability barrier. So let's go back 70 years and do that on the following slide. Let's look at this overall scheme of the lipoxygenase reaction here. A polyunsaturated fatty acid has a specific hydrogen removed by the enzyme on one face of the molecule, and molecular oxygen reacts on the other face to form the product, a specific fatty acid hydroperoxide. These enzymes occur widely in animals and plants, rarely in prokaryotes. There's usually about half a dozen different lipoxygenases in any one species. The animal ones weigh 75 to 8 kilodaltons. They're a single polypeptide. The plant ones can be bigger, up to about 100 kD in size. But still, they all contain the same catalytic structure, centered on a non-heme iron in the middle of the catalytic domain, shown here as this purple sphere. The enzyme reaction is a complete and covalently complete reaction, so they don't require cofactors, although calcium stimulates some of the enzymes. Now we can do what I was going to do, go back 70 years. So it was recognized in the 1930s and 40s that there's very high lipid peroxidizing activity in various plant sources, and especially soybeans. And by 1947, enzymologist Hugo Tiorel had purified and crystallized the main isozyme soybean lipoxygenase 1 from soybeans. And his prep actually has never been bettered in catalytic activity. It's very high catalytic activity. He could see there was 325 turnovers per second. That's a kcat of over 300. And that's really big for any lipoxygenase enzyme, because at least among the animal lipoxygenases, they can't even get within tenfold of that activity, and maybe some of them are 1,000-fold less active in terms of kcat. So 15-LOX1 can maybe do a tenth of the activity of the soybean enzyme, but some of the others are really working 1,000-fold lower rates of reaction for reasons that are not at all understood. Let me make one other point about this early characterization of soybean lipoxidase. It's actually not a lipoxidase, because oxidases are enzymes that reduce molecular oxygen to either superoxide, hydrogen peroxide, or water. So these are really oxygenases, dioxygenases in fact, adding both atoms of O2 to form the fatty acid hydroperoxide. That's a covalently complete reaction, so no reducing cofactor is required. The soybean lipoxygenase has been the go-to enzyme for years and years. It was the first to have the complete structures of the fatty acid hydroperoxide products analyzed and correctly assigned, the first enzyme to be sequenced, and the first lipoxygenase to have a crystal structure. And these are two papers that reported on the initial crystal structure of soybean lipoxygenase. Very handily, soybean lipoxygenase accepts a wide range of polyunsaturated fatty acid substrates, not just the C18 ones that we get in plants, in the beans, but C20 and C22 substrates, arachidonate and docosahexaenoic acid, are very good substrates too, which is very useful. Of course, it goes 1,000-fold faster than some of the mammalian enzymes, but one of the nice things about it, it is so stable. It will keep in a 4-degree fridge for years or even decades, so that's very useful. So it remains a workhorse for structure function studies of lipoxygenases, even though to some extent it's got atypical features. Now, I mentioned there was about half a dozen different lipoxygenases in plants or animal species. In Arabidopsis, the main substrate would be linoleic acid or perhaps alpha-linolenic acid. And the two reactions that could occur are either making the 9S hydroperoxide or the 13S hydroperoxide of those substrates. And there are specific enzymes located in different parts of the plant, many of them related to making what's really the plant equivalent of a prostaglandin in a sense, jasmonic acid. extremely important in plant growth and development and arranging plant defense. Humans have got 6 different lipoxygenases, and the main substrate is arachidonic acid, so there are more options for where the oxygen can go. For example, 5-lipoxygenase in leukocytes makes the leukotriene products. There are 2 15-lipoxygenases, and there are 2 12-lipoxygenases that make opposite stereochemistry. These top 3 occur in epithelial cells, and the top 2 are involved in the skin barrier formation, as I'm going to get to towards the end here. But first of all, let me just introduce in very general terms what the functioning of lipoxygenase enzymes, how it could be classified. Now, I made this up about 20 years ago and looked at it recently. I thought it wasn't too bad. So I've updated it a little bit, but this is the general gist. So one of the things lipoxygenases are doing is making lipid mediators. So essentially initiating signaling pathways which will end up making mediators that act on G protein coupled receptors or induce gene transcription. If we consider this, usually if you're going to make a mediator, you make one single product, one specific hydroperoxide. That's a characteristic usually of these signaling pathways. And that single fatty acid hydroperoxide after reduction might be the end product of the pathway. And in fact, there are G protein coupled receptors for 8-RHET and 12-SHET, as I'm going to illustrate for the 8-R very shortly. Obviously, forming the first product could just be the first in a chain of reactions that leads to the signaling mediator. For example, through the jasminates in plants, the leukotrienes in leukocytes, the lipoxins and resolvins, the anti-inflammatory signaling molecules in mammalian systems. And mostly they work on G protein coupled receptors, although jasminates activate gene transcription. So that's a signaling mediator. But of course, there are converting the fatty acid to a peroxide, and this could have physiological or pathological implications. And often you can think that the enzyme that does this doesn't do the reaction quite so cleanly. It actually makes perhaps a mixture of products, and instead of just working on free fatty acids, it might also be active on fatty acid esters too. So one of the well-known reactions that was peroxidation with a physiological implication was a story of the 15-lipoxygenase in reticulocytes, the 15-lipoxygenase being implicated in converting these early red cells into the mature red cells in our circulation. But then the gene knockout of the 15-lipoxygenase came along, and apparently the red cells were okay. And so this story was dropped for a while. But very recently, Hart-McCoon and colleagues have re-examined this and shown this gene redundancy accounts for the lack of actions of the gene knockout. And so this is still a sort of a real story here where the peroxidations in the reticulocytes lead to structural changes that give us the mature red cells. Of course, it can also be peroxidation has bad effects. And I don't know how things are going in the plant literature, but in the mammalian literature, the popular way to die these days is by ferroptosis. It used to be lipid peroxidation, but now we like ferroptosis. Causes cell injury and cell death. And because lipoxygenases make fatty acid peroxides, that can be part of the story. Now, a third arm I put in here has been found in plant oil seeds, where peroxidation of the triglycerides is important in seed germination and leads to the use of the fatty acids as fuel for germination. Now, these are guidelines, not rules. So I'm going to get later on to a case where we go down the 12-R lipoxygenase pathway. It's an intermediate in the pathway in keratinocytes that leads to structural changes in the formation of the mammalian skin permeability barrier. But let's get back to how these enzymes work and how their reactions are controlled in principle. So here's 3 mechanistic facets controlling product synthesis. Is to do one with the alternative orientation of the substrate as it goes into the active site, going in head first or tail first. It could be the frame shift sliding of the fatty acid in the active site. And then there's the issue of how mirror image products are controlled. How do we get 8R versus 8S stereochemistry of the product in different lipoxygenases? So let's go first to this alternative orientation, because this was worked out actually 50 years ago. It was known at that time that linoleic acid in soybeans is converted to the 13S hydroperoxide. This was shown by Hamburg and Samuelson. And they also showed that the specific hydrogen on the face of the molecule towards us was removed and oxygen was put back into the molecule on the opposite face. Well, it was also known at that time that in different plants, like in corn or maize, the main reaction was making the 9S hydroperoxide of linoleic acid, shown here. And this reaction was studied by the Vliegentaart group in the Netherlands, and they looked at the stereospecificity of the hydrogen abstraction at actually C11, this is N-8, but that's carbon 11, in the oxygenation of linoleic acid by the corn germs and soybeans. They compared the two, and here's what they found. They found that those reactions really were the same thing as each other. One hydrogen was taken off the face of the molecule as in front of us here, and oxygen was put on this corner of the double bonds right here on the face away from us, forming in the corn enzyme the 9S product, in the soybean the 13S. The difference is the fatty acid has been arranged in the opposite order in the active site. So the head to tail order of the fatty acid controls the positional specificity in this case, and the actual enzyme is doing the same reaction on both substrates. This alternative orientation of substrates is now completely accepted and is the only explanation for the very many different reactions that occur with arachidonic acid, where there are more double bonds and far more options for reaction to occur. So this has really been confirmed and is one of the controls of positional specificity, the switch in orientation of the substrate. Now here's the second one. This came about because when we moved into the animal lipoxygenases, there are more double bonds in the substrate, and one of the controls, in this case comparing 15S lipoxygenase with 12S lipoxygenase, is how far the substrate in the same orientation slides into the active site. And it was shown by David Sloan and Ariat Siegel that bulky fatty acids here in the active site were preventing the 15 lipoxygenase enzyme getting the substrate in any further than here, and we got the 15 product. But in the 12 lipoxygenase, there's more space in the active site. The substrate can enter further in, and we get, again, exactly the same reaction happening, forming 15S here or 12S here as in both cases. It's just the sliding forwards into the active site. So that was the second means of control, how far in the substrate goes. Then we've got the third aspect of stereo control, which is the mirror image forms. And I've been mentioning that plant enzymes can make the 9S or the 13S hydroperoxides of linoleic acid. And when the mammalian enzymes were characterized, they made the 5S, like the leukotriene pathway, 8S, and we just looked at the 12S and 15S. But they're all forming left-hand stereochemistry, S stereochemistry like this. Now, they can be on different positions of this substrate, but every single one of them has got left-hand stereochemistry. So what about the mirror image product? This is a completely different aspect of stereo control. Here we've got something that's the right-hand stereochemistry or R stereochemistry. And I was interested for a long time in, what type of enzyme makes that product and how does it do it. So I'm going to diverge a little bit here before we get to the answer to how this works with 4 different aspects of how I became interested in what turned out to be these R-specific lipoxygenases. I'll show you these 4. So the first one was the activity of 8-RHETE in inducing starfish oocyte maturation. And if you haven't seen this before, it is incredibly cute. Here's a picture of some starfish oocytes. From a ripe starfish, you can get millions of these, but these are sitting in a multi-well dish in seawater. They floated to the bottom of the dish. They're living cells. They're that color anyway. They're not stained or anything. And they're all sitting there like this with a big obvious nucleus, which is called the germinal vesicle. So if we add 8S-hydroxyarachidonic acid or 8-SHET to the seawater, absolutely nothing happens. But if we add 8-RHET, the mirror image form, and look at the cells 20 minutes later, they're going to look like this. The germinal vesicle has completely disappeared, the nucleus. They're reinitiated meiosis. This reaction, look at it again. There we go from that. They're just sitting there in seawater. Bingo, look like this. 1-methyladenine was the classic activator. It was then shown that arachidonic acid in large amounts could do the same thing. And actually it's because it's converted to 8-RHETE. And 8-RHET is a potent mediator of this reaction. This is what you're looking at here. Starfish oocyte maturation, the reinitiation of meiosis. Here are the agonists here. And it's a sort of classic study that was used to study cell cycle reactions back in the 80s, 70s, and 80s. Pointedly, because these are big cells, you can inject mediators into them. An injection of any of these three into these oocytes has no effect. So the evidence points to a cell surface G-protein coupled receptor for 8-RHETE. And in fact, the starfish genome hasn't been sequenced yet. And there's no indication as to what specifically this receptor is, but it's obviously there. It's a pretty nice story. So that's number one in terms of my interests. The number two was on the lipoxygenase allene oxide synthase pathway to prostaglandin-like products. This actually occurred in coral. But in fact, it mimics this well-known pathway in plant systems that lead to jasmonic acid, the little prostaglandin-like molecule of the plant world. It starts with, in the case of the plants, a 13-lipoxygenase, making the hydroperoxide, and then a specific cytochrome P450 allene oxide synthase converts this to this allene epoxide, where the double bond impinges on the epoxide. This structure here will naturally cyclize into a 5-membered carbon ring, and with a few minor changes we get to jasmonic acid, the signaling product of the plant world. Well, in corals, there was a similar pathway that we worked out. Arachidonic acid is converted by an 8-R lipoxygenase, look how it's mirror image here, to the 8-hydroperoxide, and then an allene oxide synthase that we identified as a catalase-like enzyme converts the hydroperoxide to the allene oxide, which can cyclize to give these prostaglandin-like products. They're not quite prostaglandins, but they're getting there, but not quite. And actually, the role of this pathway in lower animals, which is as far as it got in evolution, is actually unknown. But it's a sort of interesting and quite prominent catalytic activity in certain lower animals, like actually it's in starfish and in corals. So that was 1, 2. I'm going to skip 3 at the moment, which was the occurrence and role of the 8-R lipoxygenase pathway in the mammalian epidermis, and go to solving how the products of RS chiral products are made by lipoxygenase enzymes. So here's the situation again. We've got all these S-lipoxygenases. They're essentially putting the reaction into the enzyme in exactly the same way, just the substrate changes around or slides in differently, and we get S stereochemistry. How do we get the R? Well, we purified a coral 8-R lipoxygenase and showed that the enzyme is really just another lipoxygenase when you look at it. It's got all the features that you would expect, same non-heme iron, same equivalence. So here is how we switch R and S stereochemistry. And I'm illustrating this on linoleic acid, looked here in perspective. Yep, so here we have you can see that the relationship really is between the two corners of this reactive pentadiene in the fatty acid. If we react on this side here, on the face looking towards us, we get the 8R product, and 9S if we go on the other corner. And on the back face of the molecule, 13S hydroperoxide is related to the 9R. If the oxygen simply switches the side of the pentadiene that it reacts on but stays on the same face. That's the principle of switching S and R. So really, 13S and 13R enzymes would not be related to each other. Much closer are 9S and 13R, or 13S and 9R. Now it turned out, John Guido Coffa in my lab investigated the different differences between the sequences of RNS enzymes, and he eventually, not at first, but eventually spotted an alanine that's conserved in S-lipoxygenases and a glycine that's conserved in the R-lipoxygenases. And in this example here, doing the mutagenesis experiment, we're looking by HPLC at the natural product you get with a soybean lipoxygenase forming its usual 8S hydroperoxide. We get unreacted linoleic acid here. Here's the single product we get as expected. But we change that critical alanine to glycine and then make a mutant soybean lipoxygenase. It partly switches the reaction of oxygen across the face of the molecule from 13S out to 9R. So we get two products like this. And this was shown for several different enzymes by John Guido. And since then, it's been shown that it's a pretty good rule among all lipoxygenases. There are a few exceptions, but by and large, this alanine in this equivalent position in S-lipoxygenases gives us the S product, and if we have, and the glycine is in the R lipoxygenases. It's pretty amazing that that little change can make that much difference. And here's an alignment from John Guido's original paper that shows this alanine, alanine, alanine in all these different plant and animal lipoxygenases. And then in that case, there were only a few R lipoxygenases known, but they had glycine there, and he switched these in 4 different ones, and indeed it switched the reaction of oxygen across the face of the molecule. And here's a picture on the left here from the only crystal structure there is with a bound substrate. It's the 8R lipoxygenase with bound arachidonic acid. Here's a little glycine that's in this R lipoxygenase. Oxygen would approach from our angle here onto and top of there, forming the 8R hydroperoxide here. When this is switched to alanine, it partly nudges the oxygen across to the other end of the pentadiene, and we get the 12S product here. So 12S here, 8R here on the same face of the molecule, the face that we're looking at. So that was the third aspect of stereo control. So that leaves me a little bit of time to go back to the third of my long-time interests in these R lipoxygenases, which is the occurrence and role of the 12R lipoxygenase in the mammalian skin epidermal barrier. Well, 12R lipoxygenase gets its name from its reaction with arachidonic acid. It makes the 12R hydroperoxide, 12R-HPETE. But actually in the skin barrier story, the substrate is a linoleic acid ester. Actually, linoleic acid, the free acid, isn't even a substrate for the enzyme. But the linoleic esters work, and they form the 9R hydroperoxide of those esters, or the 9RH-POD esters. These are the things that control the mammalian skin permeability barrier. It's one of the controls anyway. And what is this barrier? Well, you can say it's required for life on dry land, prevents transepidermal water loss. I guess in the plant system, the cuticle performs the same function. And I put a little disclaimer here. I've got 60 slides on this subject. I'm only going to show you a few of them. But I want to give you a general idea of the story and what some of the issues are in this story. First of all, let's look at the substrate of the 12-hour lipoxygenase in the mammalian skin barrier. So linoleic acid, and it's esterified in a skin-specific ceramide, an acyl ceramide, which is abbreviated to EOS. And here it is here. Okay, the E stands for esterified. The O stands for omega hydroxy because this ceramide is omega hydroxylated up here at the end of this very, very long chain fatty acid, which is up to 36 carbons long. And this is the sphingosine part of the molecule here, which is actually quite typical. And specifically on the omega hydroxy, we have an esterified linoleic acid. And that's the substrate of the 12-hour lipoxygenase in the mammalian skin barrier. And the 12-hour lipoxygenase is just one of the genes that have been discovered to be critical for making the skin waterproof. It's actually to stop water leaving, rather as well as to stop it getting in. So these are lipid-related genes involved in the epidermal water barrier formation and showing the effects of the gene knockout in the mouse. The mice all die due to water loss when born, or inactivating mutation in humans. So these are extremely rare, but have been identified by geneticists, which is the way most of these genes were found. And the human phenotype is the babies survive, but they develop a hyperproliferative skin, a scaly skin called ichthyosis. And one can think of that as kind of like trying to rescue the defect in the barrier. The skin is leaky. They still have transepidermal water loss, and the skin tries to hyperproliferate to compensate for this. So these enzymes here, many of them are involved in making this EOS ceramide. This one here forms those very long chain fatty acids. This transporter puts the EOS ceramide into the right vesicles for transport to where they're needed. This is a specific acetyl-CoA synthetase that forms the CoA ester of this now omega hydroxy ultra long chain fatty acid. This P450 forms the omega hydroxy group on the ceramide. This actually seems to be a facilitator of making EOS synthesis. This enzyme here esterifies linoleic acid specifically to form that acyl ceramide EOS. Then we've got the pathway enzymes that I've been looking at. For some reason that we're trying to figure out, you need to oxidize the EOS ceramide in order to form the skin permeability barrier. And the first step in that pathway is the 8-hour lipoxygenase, as we said, acting on the linoleic ester, making the 9-hour hydroperoxide. Then this enzyme here, epidermal lipoxygenase 3, is an unconventional lipoxygenase that isomerizes the 12-hour lipoxygenase product and makes an epoxy hydroxy derivative. And then the recently studied enzyme that Val mentioned is this SDR9C7 short chain dehydrogenase. It takes this epoxy hydroxy product and makes the epoxy ketone. Now we knock out any of these enzymes individually. The mouse pups die of dehydration within hours of birth, and the humans, in those rare cases where they have the mutation that inactivates them, they get this ichthyosis hyperproliferative skin. Here's the case in the mouse. This happens with knockout of any of these, but actually I'm showing it for the 12-hour lipoxygenase knockout, but you get the same thing, for example, with the epidermal lipoxygenase 3 knockout. Neonatal lethality due to transepidermal water loss. The skin can be shown, this is a wild type, can be shown to exclude dye normally, but in the knockout, the skin is permeable and can be seen to take up the dye, which it shouldn't. After birth, the wild type enzymes, and this is in hours here, up to 6 or 7 hours, the wild type pups maintain their weight because they can keep the water in. But the 12-hour lipoxygenase knockouts, ELOX3 knockouts as well, the SDR knockouts also, They lose weight over a few hours because they're losing water, and they die of dehydration within hours of birth. This has been particularly studied by Peter Krieg and his colleagues in Heidelberg, and they showed this for both 12-hour LOX and for the ELOX3. And the babies, in these rare cases where they have the inactivating gene, they survive. They're born with something that's called a collodion membrane on them, which doesn't look very good, but they do survive, and within months they're starting to get this hyperproliferative scaly skin. And they're stuck with this for life. It's an incurable congenital scaly skin ichthyosis, being named after the Greek word for fish, as in fish scales, fish scale-like skin. And figuring out what's going wrong here is our whole emphasis. Why is it that we need to oxidize these lipids to make the skin properly intact? Well, the pathway side of it we have worked out over the years. First of all, Bill Boglin cloned the 12-hour lipoxygenase and showed its activity. Jiong Yu here showed the activity of ELOX3, that it's on a hydroperoxide isomerase. So here's the linolate ester, the ceramide, going to the 9R hydroperoxide. ELOX3 converts this to the epoxy hydroxy derivative. And Ushan here showed that these reactions happen on EOS ceramide using the recombinant enzymes, and she found those specific oxidized products in normal skin and absent in the skin of the 12-hour LOX knockout. So we know these reactions occur on EOS ceramide and are, for some reason, super important in keeping the skin intact. Further studies showed that this epoxy hydroxy product is hydrolyzed, we now know by the epoxide hydrolase called EH3 or EPHX3 is the gene, to a specific linolate trial. This was shown by Chris Thomas when he was in the lab, and Takahito Chiba as well as a postdoc. And Haruto Yamanashi showed that the activity with the recombinant enzyme of ELOX3 best matched this specific transformation here. But more recently, we've realized that this is maybe not an activation step in the pathway to the skin barrier, because Matthew Eden and Daryl Zeldin at NIEHS have the epoxide 3 hydrolase knockout mouse. And this has got a very mild barrier defect that doesn't match what happens with the other gene knockouts. So it's thought that maybe this is a pathway that fine-tunes the pathway, but it's not the main thing that's critical in making the barrier. Now, let's get to what is wrong when you don't oxidize these lipids. Well, let me say this. We know the oxidations happen, and actually I can show you, we know what's wrong structurally with the barrier. What we're still working on is what's the connection between doing the oxidations and getting the barrier intact. But I can show you that what's wrong when you knock out any of these enzymes is there's an elimination of covalently bound ceramides in the skin barrier. We can see this as an end result. We're still not sure how we get there. But this is my cartoon of the whole thing. So on the outer layers of the epidermis, the corneocytes are dead cells, but they're fused together to form this barrier. And the fusion involves differences both inside and outside the cells so that they're absolutely watertight as a seal. So if we look at this area between two of these corneocytes with the electron microscope, can you see this shadow here? That's inside one corneocyte. And then in the lower one here, there's another shadow like this. This is what's called the corneocyte envelope, and it's a layer of polymerized protein that's replaced essentially the plasma membrane. So they don't have a plasma membrane anymore. That corneocyte envelope is around the edge of all the cells. And then in between the cells, there are these multi-laminar layers of lipids and amino acids forming a structure like, actually looks like a plasma membrane, except it's many, many layers thick, and it's got cholesterol and these ceramides in there. And this is in between the cells. And so I've cartooned that here. Here's our corneocyte envelope, the polymerized protein in one cell. Here it is in the other. Here's the extracellular lipid lamella. So that's between the cells. But between these two parts of the barrier, we have a layer of covalently bound ceramides, covalently bound to the corneocyte polymerized protein. And when we knock out the 12-hour-LOX or ELOX3 or this SDR enzyme or many of the previous enzymes, what's missing are these covalently bound ceramides in the structure. And you can imagine that they actually form a scaffold between the polymerized protein and the layers of lipids here. And when they're gone, the barrier structure is lost. This covalently bound ceramide layer is called the corneocyte lipid envelope. And it's composed of EOS-like ceramides covalently bound to the protein coat. And that forms an intact barrier, and the covalently bound ceramides are missing when the lipoxygenase is knocked out. So here's what we found very recently in collaboration with Professor Akiyama and colleagues in Japan. They had found this, first of all, human subject who had this SDR9C7 short-chain dehydrogenase inactivated, and they prepared the mouse knockout. And the mouse knockout had the usual phenotype that the pups die of dehydration within hours of birth. And so we could see now, so we worked on this with them and looked at the skins of normal mice and the knockouts, and we could see what was happening. That actually, look down below here, this epoxy hydroxy epoxylin still in the EOS ceramide is converted to the ketone by this SDR9C7 enzyme. That's an NAD-dependent reaction. We could see this in vitro with the recombinant enzyme, and we could see the products in vivo when we look for the keto-containing epoxy ketone in the ceramides. And this pathway here, going from EOS to the 9-hydroperoxide to the epoxy alcohol to the epoxy ketone, knock out any of those steps, you eliminate that covalent protein binding, and the barrier is done for. So we're still working on why is that. And this is at least this is a cartoon suggesting one possible reason that it could be involved. Here's the acyl ceramide containing linoleic. So this is the red part of the ceramide part, and this is linoleic acid. You can see it's 2 double bonds here, esterified in EOS ceramide. The lipoxygenases, 12-hour-LOX and ELOX3, convert this to the epoxy hydroxy derivative. SDR9C7 converts the alcohol to the ketone, giving us this unsaturated epoxide ketone. And this is known to be a reactive structure, which even non-enzymatically can react with protein to form covalent double bonds. So one of the possibilities here is that this formation of this epoxy ketone is critical because it allows covalent reaction with this polymerized protein, the cell envelope as it's called, the CE. And this is a working hypothesis, if you like, at the moment. When you knock out the SDR9C7 enzyme, in people you get ichthyosis, the mouse pups with the knockout die, and instead of, but when they're born, they still have a side pathway hyper-exaggerated. Now we see that there's EOS trihydroxy there in 100-fold higher amount than normal. when this is knocked out and we have lack of covalent binding and the mouse bucks die. So that's where we're sitting at the moment in terms of this story, trying to figure out what specifically do these oxidations do in order to get covalent binding of the ceramides and an intact barrier structure. That's still to be determined. Well, that's the finish there. I just want to thank my lab mates over the years. I'm just listed the ones here that are involved specifically in this 12-hour lipoxygenase product: Bill Boglin, Bushan Zhang, Jiong Yu, Takito Chiba, Haruto Yamanashi, and our many collaborators, including Klaus Schneider at Vanderbilt, Peter Elias, who's done electron microscopy on some of our knockouts. We collaborated with David Bayer and Jennifer Moran in looking at their 12-hour lipoxygenase knockout. Some of the work in characterizing the trihydroxy products were with Val and Chris Thomas. Then Akiyama and colleagues, we've worked on that SDR enzyme, the most recent step in the pathway. And with Matt Eden and Daryl Zeldin at NIEHS, we've characterized epoxide hydrolase 3 as forming those trihydroxy products. And many thanks for NIH funding over the years, currently by this NIGMS grant, which we're very grateful for. Okay, Val, that's that. Thank you.
Thank you, Alan. That was excellent. We have a load of questions. I've got a load of questions too, but I'll hand over to the floor to take some questions. So first of all, Ed Dennis, you have a question for Alan. Go ahead. Just waiting for Jeff to unmute you.
Throw me a beach ball, Ed.
Go ahead, Ed.
Beautiful presentation. I was particularly curious about which of these lipoxygenases can attack fatty acyls in triglycerides and phospholipids. You mentioned triglycerides near the beginning, didn't say much about phospholipids. At the end, you're talking about ceramides, so clearly some of these work on the ceramides, but they tend to be more extended. So are there some generalizations and some specifics about when lipoxygenases can work on complex lipids?
Yes, Ed, I think I would take you back to the orientation of the fatty acid and the substrate in the active site of the enzymes. Some of them go in tail first, some of them go in head first. So you can automatically eliminate any of the enzymes where the substrate goes in head first in terms of metabolizing a very large ester like a phospholipid. So for example, that cuts out 5-lipoxygenase in the mammalian system. It just couldn't happen. It similarly would eliminate the 9S hydroperoxide in the plant system. That leaves us with the ones that go in with the fatty acid tail first, and at least conceptually could be substrates, even though they've got a large ester attached to the carboxyl group. And among those, I would say the ones that like 15-lipoxygenase 1, as it's called, is certainly the enzyme that's supposedly involved in the reticulocyte story, and actually does seem to be a real issue there. And even 15-lipoxygenase 2 in the mammalian systems, which very specifically forms the 15-hydroperoxide, that will work with phospholipids, although they're not very good substrates. I think I can leave it there.
Yeah, I mean, in relation to that, if you have macrophages come into the skin, can the 15-LO1 oxidize the ceramide with linoleate attached to it? I mean, is that outside the cells or inside the keratinocytes, or where is that substrate?
Yeah, well, I don't think macrophages could get to it. As you know, the skin epidermal layer has a basal cell layer which goes up through multiple layers of development up to the top layer where we have the barrier. And EOS is being synthesized during those developments throughout the epidermis. So macrophages are just not going to get there to test it. I don't think that's been tested in vitro actually.
So
but
during some sort of chronic inflammatory situation, I guess I'm thinking about. A chronic inflammatory situation is what I'm thinking about.
Right. Yeah. Okay. Then when the barrier's disrupted and cells are coming in, as in psoriasis, for example, then that would be a possibility. That's for sure.
But I ask in vitro, perhaps, where it's been studied, the soybean lipoxygenase has been demonstrated clearly to work on arachidonic in the 2 position of phospholipids. In vitro, as your earlier part of your talk, have any of those others, like the 15 ones that you were discussing a second ago, have any of them been shown in vitro to actually get in and do that reaction?
Yes, 15-lipoxygenase 1, 15-lipoxygenase 2, and the 12S lipoxygenase, both the platelet type, if you like, which is a pure 12S lipoxygenase, and the rodent 15-12 lipoxygenase. All of those have been shown in vitro to metabolize phospholipid substrates.
Okay, thanks. So Chris Thomas?
I can send you the references.
I'm sure you could.
And I'm sure he will. Chris?
Hi Alan, how are you?
Aye.
You okay? So thanks for showing that picture firstly. I think it was my first day at Vanderbilt. I looked about 12. But yeah, thanks for that. Yeah, I guess my question leads on a little bit from that. You mentioned the different structural sort of entrances of the enzyme, and I was wondering if 12-R-LOX and E-LOX3 are sort of structurally different around the active site to allow that massive ceramide to sort of be anchored in any way while the reaction goes on, or do they look very similar to some of the other LOX enzymes?
Well, the short answer to your question is that they're very, very similar by alignments with the lipoxygenases whose structures are known. The same key catalytic residues and so on. However, both of them, both E-LOX3 and 12-R lipoxygenase, have an insert between the beta-baryl domain and the catalytic domain, an insert of about, in one case, 31 amino acids, another case, 41 amino acids, which forms another little mini structure on each of those enzymes. And the role of that is completely undetermined, but it's a feature only of E-LOX3 and 12-R lipoxygenase.
Do you think that would be big enough then to hold that very long chain ceramide? Because as you said, we're talking maybe up to 36 carbons in that long chain ceramide, aren't we?
Sure. Well, we're thinking, I couldn't tell you the answer to that question, but again, the substrate has to go in tail first. So really, it's only the tail end of linoleic acid itself which is entering the enzyme. How the rest of the molecule could be interacting with the outside of the protein is anybody's guess at the moment.
Okay, brilliant. Thanks.
Okay. Thanks, Chris. Jeff Peterson.
Hey there.
Is Jeff there still?
His picture is.
Hi, can you hear me?
Yeah, we can hear you now, Jeff. Go ahead.
Thank you very much for that talk. I just had a question. You were focusing on arachidonic acid as a substrate for lipoxygenases in mammalian cells, but I'm just curious about other PUFAs, linoleic acids, and I have a particular interest in those fatty acids with conjugated double bonds. So I'm curious if you could talk about other classes of substrates for lipoxygenases and their biological roles.
Well, of course, I was talking about linoleic acid and the skin barrier story. It's the only polyunsaturated fatty acid that's available and that is used in that EOS ceramide. It's pretty interesting, actually, because although arachidonic acid cures essential fatty acid deficiency, and the scaly skin you get with essential fatty acid deficiency, it cures it because it's converted back to linoleic acid. Only linoleic acid works in that skin barrier, mammalian skin barrier. As for substrates with conjugated double bonds being substrate, if you take, for example, conjugated linoleic acid, it wouldn't be a substrate. It's Yeah, that's the short end of it. You need to have the CH2 between two double bonds in order to be an effective substrate.
So the chemical mechanism would be entirely different.
Mm. Yeah.
Well, you could draw a reaction with the conjugated system, but it's unlikely to happen. Hydrogen abstraction requires more energy, and it isn't effective with a non-heme iron lipoxygenase.
Thank you.
Emanuela Camera. Oh, we're waiting.
Yes.
You're there. Great. Okay.
Yes, thank
Go
you.
ahead.
Thank you for picking my question. Yes, I was interested to know if there is like a gradient of expression of these enzymes, and if there is any effect of the oxygen tension on the activity.
Could you say the first part of your question again, please?
Yeah, if there is a gradient of expression and activity of lipoxygenases in the skin as the skin undergoes a complex process of differentiation.
Mm.
Yeah.
Well, definitely there is, because if you take mouse or human keratinocytes in culture, they are completely undifferentiated and they do not express the 12-hour lipoxygenase or ELOX3, and they don't even make EOS ceramide. Now, in principle, you can induce differentiation, it's been said, by adding calcium, but that still isn't enough. You need to put in many differentiating factors to the extent where in vitro, the cells, rather than being in a monolayer, start to pile on top of each other. Only at that stage in vitro do you start to see formation of the lipoxygenase enzyme expression, of the lipoxygenase enzymes and the synthesis of those ceramides. It doesn't happen in the basal layer. So in your question, yes, synthesis is happening throughout the epidermis.
Sriparna Banerjee.
Hello.
Hi there. Thank you so much. It was a wonderful talk. The question that I had, actually, you've given a partial answer to it from another previous question, but the substrate that you've described, the acyl ceramide with linoleate, it's very, very large. So would any other lipoxygenase substrate enzyme be able to accommodate a substrate that large?
Well, again, this could only happen if there was tail-first entry of the fatty acid into the active site, and then you've got the rest of it sticking outside the enzyme. So in principle, it would be possible for a lipoxygenase that accepts the substrate tail-first into the active site to do a reaction, although I don't believe that's ever been shown or tested. And I might say that working with that substrate with its 60-odd carbons in vitro is really difficult because it is very insoluble in aqueous solution.
Yeah, I can imagine that. Thank you very much.
So Alan, I wanted to ask, since ichthyosis, it can be caused by multiple single gene mutations, is there consideration towards CRISPR-Cas9 approaches to try and cure this in humans? Because it's quite an accessible organ. Obviously, it would be easy to deliver, I guess. But
well, I guess it could be delivered to one spot. I don't know if you can do the whole organism. They say the skin is the biggest organ in your body, or one of them. So I'm not sure how practical that would be. I'm sure it's being thought about, though, but we're not there yet.
Yeah. Okay, so Binks Wattenberg.
Hello.
Hello.
Hi.
Go
Thanks.
ahead.
That was just a fabulous seminar. Thanks so much. My question, I don't know, this is a little bit off the path a little bit, but as you probably know, ceramide is incorporated into a lot of skin creams. And in my understanding, actually also has been clinically shown to show improvement in some skin diseases. So what I'm wondering is that ceramide metabolized, you think, to EOS and to farther down the line? That is, do you know whether these enzymes are present?
I can answer that question. You know, the chemical synthesis of EOS was first reported by a cosmetic company in Japan, but the synthesis was so difficult that it just didn't become a commercial product. So the skin cream ceramides don't include the esterified linoleic acid additive, well, not additive, but, you know, covalent molecule. They don't have acyl ceramides in those skin creams. So the answer is no, they couldn't be metabolized the way we've been talking about. Hope that answers your question.
Right. But can they be metabolized to be acylated? You know, can they undergo, you know, do they get turned into EOS?
That's a good question. I have no idea. I don't know. But in principle, that might be possible.
All right. Thanks.
Okay, so we're nearly, well, we are pretty much on time, but I just want to ask one last question, Alan, I guess. I mean, the soybean lipoxygenase is a great enzyme for people like me who are interested in working with oxidized lipids, especially because it can oxidize anything and it keeps going for ages until it runs out of substrate, unlike the mammalian ones. You know, which suicide inactivate, etc. And but you know, it's really the only one we have, the only tool we have that can generate a really specific project in a really simple, easy way. And it would be great to have many more enzymes like this, because we have a real dearth of standards. And you know, they're hard to make. You use air oxidation, you get a great big mess that you got to clean up. And I know you're well aware of all the issues. Do you think that there's easy ways, or are there ways? Have we thought enough about doing more mutants to try and cover more of the mammalian physiological products in ways where we can use enzymes to generate specifically the products that we want for our, you know, biochemical studies?
Right. Well, as I mentioned briefly, the human enzymes, you just don't have the stability that you have with the soybean enzymes. So that's just one big difficult issue in terms of, you know, using them to make lots of product. You need lots of enzyme. They've also got much less catalytic activity. It's actually slightly surprising the soybean enzyme hasn't been mutated to make anything other than 9 and 13 linoleic acid products or 15 products from arachidonate. In principle, you would have thought that might be possible, but it hasn't been achieved. But yeah, it's a difficult issue because of the lack of stability of some of the enzymes that we would like to have.
Do you think the approach of trying to mutate that one has been taken and it just hasn't worked, or it just hasn't been taken enough and it should be revisited again, perhaps?
You know, logical mutations
absolutely,
have been made,
and
insights have been gathered from that. So I don't want to say it hasn't been done, but it just happens that it hasn't cottoned on to being, you know, giving us useful new products in large amounts. Let's put it that way.
Yeah. Okay. Well, maybe we should go back and think about that again, I guess. Okay. Anyway, that's been fantastic. We've run out of time, unfortunately. Any remaining questions, please feel free to email Alan. I'm sure he'd be more than happy to answer by email. But I want to say thank you again, Alan. That was fantastic. I'm always delighted to hear about lipoxygenases in detail, since I work very closely in this field. And